lung. I'm very happy to answer any technical questions on immunology in that respect. I made this mostly a non-technical, inspirational kind of talk, where I think we can use some interesting things with the science we have to prevent something like this from happening again. And I'm also the founder of a tissue engineering company right now that has technology that could support this, so full disclosure.
Right, well, I'm incredibly happy to have, yeah, I'm very, very happy to have Melanie on here. She has been I guess on Foresight's radar for a really, really long time. And we have been trying, at different events to connect and to get her here. And I'm super, super happy that someone like her now took on COVID-19. I'm incredibly hopeful about the work that you guys are doing. I can't wait for what you have in store now already, and then how it's going to evolve over the next few weeks. Thank you so, so much for joining. I'm super eager to learn what you have to share with us this morning.
Yeah, absolutely. If everyone's feeling a little shy, we could go ahead and get started, and then maybe discussion will be prompted. What do you think?
Sounds good. Go ahead. And then afterwards, if people want to chime in and have questions, maybe they can then introduce themselves and their background so that we know the relevance.
Okay. All right, great. I'm going to share my screen, you guys. And this is always a little scary because I have 18,000 windows open at once. But here we go. All right. Is it going full form on your guys' screen? Or does it just, okay, great. Okay.
Well, no, we're not seeing the display mode. We're seeing presenter mode with all the slides.
Let me take off my other monitor. That might be the problem. Let me see if we can redo that.
Yeah, that's better.
We had, yes, perfect.
There we go. Okay, great. I have my email here if you'd like to have further discussion or for me to dig up all the references that I used in putting together this talk. I apologize, I ran out of time to put all the references on each slide. But I'm always happy to share my library of research with anyone who's interested. So like I said, immunologists who studied three-dimensional tissues as a physical network of independently moving cells. There's a lot of good modeling on how they move, how they respond to chemokine responses, and it's a system of information transfer. Once a barrier is breached, which we'll talk about a lot, you have a system of information transfer that is cascaded all the way out to an antigen-specific response, which is something you would need to prevent or reduce viral infection. Okay. And so this is a very high level talk. Again, stop me anytime for questions. I'm very happy to share what I know. Okay, so here are my goals. I want to review our current approaches to plague management. I think a lot of people have been thinking about this. Feel free to chime in if I've missed anything. What does your immune system do when you're infected? I'll give a high level overview of something like that. Why and how I believe antibodies actually offer this rapid solution for an emerging pandemic disease. I think that's really important, and it's a way that I believe we can actually get ahead of these things so we don't ever have a disaster like this again. And with that, that's my novel way to approach a plague prevention. I'm going to float some kind of high-level ideas there and how I think about this. And I want to contextualize kind of the toolbox that we have against SARS-CoV. There's a lot of different ways we can approach this, but as we all know, timing is everything, especially when something spreads this quickly. Okay, this is kind of my doom and gloom slide. I really do believe our world has been changed forever, and it happened quite quickly. It happened faster than we were prepared to respond to. This is the greatest pandemic in living memory, and it's not going away. And just a small plug for an article I wrote about why I think that's true. This is on Medium. You can find it under my name. Feel free to read, comment, give feedback. This is me, a lot of ideas last Monday when I wrote it. Okay, let's talk about the immune system. There are three branches of the immune system. We have a barrier branch. This is how it's taught to us in undergrad, grad school. This is how I taught med students at UCSF. You start with a barrier, you have an innate response, and then you have an adaptive response. These can be thought of as having a bit of a Venn diagram around them. A lot of the barrier/innate responses coexist. A lot of the innate/adaptive responses coexist. That transfer of antigen, once that barrier is broken, is what I was talking about earlier. The innate system talks to the adaptive system, which takes a little longer to catch up. And that barrier in that innate system, that is what's there to give the adaptive immune system time to catch up, which has a lot of parallels into how we as a society tackle a viral pandemic. Okay, so one of the things I proposed in this article was that I think that our society can be thought of as a different branch of the immune system. And if we are able to hack the immune system to develop adaptive responses ahead of viruses, we can do something called adaptive therapeutic development. All right, so this is a standard immune system response and how I think we can hack it to do better than we have. And let me see, sorry, I'm gonna read a slide here. Yeah, so each one of these systems, as I mentioned on the prior slide, is a way for the immune system to buy time to neutralize a pathogen. So physical is a barrier, that's our skin, that's the high pH in our stomach, that's the intestinal wall lining that is just packed with immune cells. And if a pathogen can't reach those barriers, it can't infect you. Mucous membranes, the cilia in your nose, your lung, your throat, all of those are designed and are effective against many pathogens. So that physical barrier works pretty well for a lot of pathogens. Some pathogens, however, have evolved to get past that, or you cut yourself and bacteria gets in. Also, the physical barrier will sequester pathogens. I'm sorry, I didn't mention that, but it will actually form a capsule around some pathogens and prevent them from entering the rest of your system. That is also facilitated by the innate system, which is a broad response mechanism. Innate is why you have pus in your finger if you have a bacterial infection. A lot of cells are going there. They know there's a danger signal, they're responding, and they release a lot of different cytokines and different mechanisms. Neutrophils have things called neutrophil nets. They kind of die on site, that's what the pus is. But it's a broad response. It's not targeted to a particular pathogen, antigen, bacterium, or whatnot. And it will directly involve killing, the cell will go in and directly kill an infected cell. And some of that, again, like I said, this is kind of a Venn diagram, will overlap with adaptive cells. Adaptive immune response is a specific pathogen-targeted response. It takes weeks to develop one. This is how our vaccines work. You get an antigen that's specific to a viral pathogen. You have a shot in the arm. A couple weeks later, you're fully protected. And that's how that works. Do you guys have any questions? Did I miss anything?
I have a question. Can you hear me?
Yeah.
The innate system, I mean, I'm familiar with some of this stuff, but it's been 15 years, yeah. So remind me.
Sure.
How does since the innate system does not target particular pathogen-specific molecules, what does it target? Does it target damaged cells generically in some way? Like, how does it know to go to the site of the infection and what to kill?
Right. So, and I'm getting a signal, my internet's unstable. I'll wait for a second. Okay, back on. So I should say that when I say pathogen specific, I'm referring to signatures that are specific to a pathogen. So when you think about influenza, we say H1N1, H5N1. Those are specific to cellular proteins that will mutate and show a different signature. However, influenza, like many pathogens, our bodies know that we don't typically have single-stranded RNA floating around. And so that is a viral signature that our bodies will respond to that isn't specific to the virus per se. It's specific to a molecule within the virus that is typical of many single-stranded RNA viruses. There's another one called CpG DNA, and so there's a different methylation pattern on many viral DNAs. Our bodies are innately built to recognize, hey, there's a pathogen here. We don't know exactly what the signature is, but we've got some foreign DNA. What we're going to do is we're going to go kill everything site that has that foreign DNA around it and have a cytokine response that alerts the adaptive immune system that we've got something new for you to look at. And so those are called viral signatures. PAMPs is their shorthand, PAMP. Those are pathogen-associated molecular profiles or patterns, rather. And so I'm happy to share recent literature on that, but is that clear?
Yes, that's wonderful. Thank you.
Okay, awesome. All right. So moving on, when the adaptive system does fail, the pathogen will persist. And an example of this that we might all be familiar with is HIV persisting in our population. We can make antibodies against HIV. It's not that the immune system doesn't recognize it. What happens is we have a failure of the antibodies to neutralize the pathogen. And so HIV has viral surface proteins, much like other viruses, but their distance is so far apart that the antibody, which is forked, can't bridge that gap, and it doesn't form a good neutralizing bond. And so when the adaptive system fails, we have viral persistence in the community.
I have another question.
Absolutely.
So forgive me if you're already going to talk about this later, or if it's too much and it's you got other things. But I've always wondered, or I've been wondering as I try to remember my immunology, the adaptive system, as I recall, it kind of has two different things that it does, right? It makes antibodies for the particular pathogen, and it makes specialized killers or specialized T cells that can make killer cells attack pathogen infected cells. Okay, can you talk about those two things? Great.
Yeah, absolutely. So that was my next slide. Well done. Okay. Thank you. You can write my talks. All right, so cellular, there are two branches to the adaptive immune system, which Pion just referred to. This is Immunology 101. Once this information is passed along the cells that can develop a very specific response to a particular pathogen, you have cellular response. These are T cells, NK cells, or rather NKTs, sorry, NKs are more of the innate. That's my mistake there, but you have direct cellular contact between these T cells, dendritic cells, different branches and types of T cells exist within that milieu. That's a whole other branch of immunology. And then you have a humoral response. This is generally relegated to B cells, and humoral is a reference to human blood. It's one of the 4 humors that Hippocrates, the father of medicine, modern medicine, identified. And so humoral refers to the fact that these B cells, once they undergo a series of, and I don't have a slide on how antibodies are generated, but so happy to describe it. B cells undergo a series of guess and check steps, and they, in that guess step, they're randomly mutating something called the VDJ region. There's CDR of the antibody is what it ends up transcribing to. And that is an area that random somatic hypermutation occurs in at a higher rate than anywhere else in the human body. And it happens in response to the B cell has done its first round of mutations back, and it talks to local antigen presenting cells and T cells, and it says, did I make the right mutation? Did I produce a productive antibody? And if it has a stronger binding strength than the other B cells in its neighborhood, it gets all the possible signals to go back and divide again. This is how antibody evolution occurs. And so that guess and check step happens many, many times until you have a dominant B cell population that is expressing and producing a very strong, significant antibody. And after a certain number of times of guessing and checking, those B cells become plasma cells, and they're plasma B cells. And what they do is they hunker down in the tissue where they are. They expand to be this really large cell that's basically a bag of antibodies, and they start producing it and secreting it into the blood. And so once you have that response, your system, your plasma is just chock full of specific antibodies. And so that is how it's named, and that's part of why it's named the humoral system, is because it's a lot of circulating antibodies, not necessarily circulating cells, but circulating proteins that are virus specific or pathogen specific. You guys have any questions? That was quite a lot. Okay. All right, so that's how we're going to produce an antibody. That's how people around the world are producing antibodies right now.
Wait a minute. Could you go back? Could you go back?
Yeah.
Are you going to cover the right-hand side later?
Oh, sure. I can talk a bit about the cellular response.
Yeah,
that's
so
not a problem.
I want you to do that if you don't mind. And then I also would like you, could you mention the, seems like there's an arrow connecting the two sides, but also what are the time frames for these two sides? Are they the same or are they different?
They're a little bit different. So the T cell, dendritic cell, that kind of response tends to overlap a bit more with the innate response. So NKT cells and T cells can directly kill virus-infected cells. So a version of a T cell is called a CD8 positive T cell or a cytotoxic T cell. These are the T cells that are used in cancer immunotherapy because they can go out, their little receptors will recognize a cell or a protein on a cell. And once they have that recognition and that contact, they'll kill that cell directly. And that's why they're so powerful in cancer immunotherapy. You change their receptor to recognize the tumor cell and they kill that tumor cell. Typically how it works in the body is they recognize a pathogen or a pathogen infected cell rather, and then they kill that cell directly. So those are called CD8 T cells, cytotoxic T cells for short. And they are the responsible ones for cytokine storm. Cytokine storm is produced by the adaptive immune system, and it is actually what causes patient demise in situations like this. And so people who can't breathe and are having extreme inflammation, edema, etc., it's because of the cytokines that your body has released. It's not necessarily viral infection. And so one of the primary strategies in viral infection is actually reduce inflammation as much as possible to give the other branch of the immune system, the B cell branch, time to catch up. There is an arrow over there because there's another type of T cell, the adaptive B cell response. And so they do this with a different set of cytokines that are not there's some overlap between them, but Venn diagram, mostly a different set of cytokines that support B cell development, class switching, which is seen with somatic hypermutation. They can also directly present antigen to B cells. I was part of a lab that produced a paper on that, directly demonstrating T-B cell contact. There's a Miller et al., Miller-Cahalan et al. From I think it was like 2005 maybe, that shows before. And then I believe it was 2007, I was the lead author on a paper that demonstrated that with different responses to B cells. So you can either induce an immune response or you can actually induce tolerance. And so that was a mechanism we were looking at to induce tolerance in autoimmune disease. So those T helper cells go over and they talk to the B cells directly. They also interact with antigen presenting cells, the ones that kind of gobble up, neutralize the virus, chop it into little bits, and then show the immune system the information. And so that's kind of a triumvirate of cells, antigen presenting cells, T helper cells, and B cells that drive the antibody response and support the B cells in their class switching. All right, again, that was quite a lot, guys. Any questions?
Also, feel free to raise your hand with
there
the
are
Zoom
no dumb
feature.
questions here. I've never.
Yeah, feel free to raise
Also,
your
I
hand in
can't-
the Zoom feature
can't
if you want,
see
and then
all
I
you,
can monitor.
Yeah. Thank you, Allison. Yeah, I can't really see everyone because I have my slides up while I'm talking. Okay. Yeah, and happy to talk more about that. These are very well researched large areas of immunology. There's many subsets of each of these types of cells, and yeah, it's extensive. But that's generally what's going on there. Okay. All right. So having studied the immune system for quite a while, I believe that the power of human antibodies has not been leveraged in a way that we could for treating pandemic disease. And this is why I'm here talking about this today. Antibodies literally, they buy us time. And how we know this is a few things that we can point to. Just antibodies confer. It means you haven't had a full response to a pathogen. You've never probably been exposed to a pathogen. But if you have an antibody that recognizes that pathogen, you can prevent infection. It a priori starts neutralizing that virus before you have systemic infection, and maybe you get a small innate response, kind of killing a few leftover virally infected cells, but you truly do have protection. And there's two places that we see this naturally. Well, there's one place we see it naturally, one place where we use it quite often for people. So the place where we see it naturally is passive immunity conferred from mother to child during breastfeeding. Human breast milk is full of antibodies, and it's full of antibodies that are being made by the mother during her recent exposure or because of her recent exposure to the local environment. Whether pathogens are in the local environment, she has a high concentration of antibodies against those. That's happening in the background with our immune systems all the time. So that child who comes into the world with a very naive immune system and really not much of an adaptive immune system quite yet, they are given, however long you're breastfeeding, they're given protective immunity or passive immunity to the local environment. And so this gives them time to develop without getting sick. And so that's where the idea of using transferable human antibodies really came from. It works quite well. And
Question.
so the way, yes, of course.
Two questions actually. One is, so do antibodies in general just target like the raw virus when it's outside of a cell? And if so, what good is an antibody once the virus gets into a cell, or does it just leave that to the T cell system? That's the first question: is are antibodies only good against viruses that are outside the cell, and then or bacteria or whatever? And then the second question is, how does the fetus not get any antibodies that are directed against its self proteins, because it has different proteins than the mother?
Yeah, those are really good questions. So first, antibodies are generally only good towards virus that is free floating. They're quite good though at neutralizing it. And once you get virus into a cell, it's true the virus will start to reproduce in the cell, but this becomes really a numbers game. And so you do have the innate system that will play catch up, but if you've neutralized a vast majority, and these antibodies are quite small, they can go all over the body. If you've been able to neutralize the vast majority of the virus, your innate system can really take over from there. That's just, I believe there's a numbers game in there. I don't know if anyone's investigated exactly what it is. We know that different concentrations of antibodies work in different ways. Prophylactic antibody treatment against anthrax, they give you about 1 gram of it. Other antibodies, it's milligrams. So there is there's a numbers game in there that depends on the antibody, the rate of infection, the timing of infection. But if you a priori have a lot of circulating antibodies that can neutralize the virus, it does seem to work. And I believe anytime you do have a breach of that, it's so minor that the innate system can play catch up, and does it so well that you don't often, that I've found in the literature, you don't often even have an adaptive response. It doesn't even get that far. So, and there's some mechanisms that can kind of hedge in favor of antibody neutralization. For example, if you get a light tickle in your throat, you felt sick for maybe half a day, that may have been an antibody neutralizing it. But that light sickness, that tickle in the throat, causes local minor edema that can collect more serum antibodies there too. And so that's kind of a more barrier function, but that can happen. Your second question was, sorry, I've forgotten it. Creon, would you mind repeating it?
It was, how is it that the fetus is protected against the mother's antibodies that might be targeted against the fetus's proteins, which say are from the father, which are foreign to the mother?
Right, so fetuses aren't. So we have, the immune system has a lot of balances around developing autoimmunity. We generally reduce autoreactive cells in our thymus as we're growing up. And autoreactive cells are often, they're either reduced or eliminated, the genetic sequences that respond to self-proteins. And we have a lot of what they call housekeeping genes expressed in the thymus. And so as T cells are developing in the thymus, they see just this wide variety of standard human proteins. And any T cell that responds to them goes quiescent or is killed. In the periphery, when you develop spontaneous autoreactive cells, often what happens is the immune system will shut them down. We have something called regulatory T cells. It's another type of T cell. And regulatory T cells will stop autoreactive immune responses to the point where people are looking at them as a therapeutic for for reducing autoimmune disease. There was a company just finished their A round, Sonoma Biotech, and it was founded by my prior PI at UCSF, Jeff Bluestone, and he's looking at using regulatory T cells as an autoimmune therapeutic. They actually stop immune responses. However, to your question, your point, fetuses are not protected. And so mothers can have responses against the baby in utero. And this is, you know, a lot of sad stories. This is a lot of the reason why you get spontaneous miscarriage. A lot of people may know that there are blood types involved in this. If a mother is O negative and she has the father is O positive, she has the potential to be primed to be reactive against that positive Rh type on the surface of a blood cell. And so often these mothers have extreme difficulty conceiving again because the immune response will reject that positive blood type. So yeah, so that's pretty well known. It's not that it's protective, it's that we generally don't have autoreactive responses. Is that clear? And I think there's a lot more research there that I'm glossing over. Okay, great. Okay, so antibodies are very, very powerful. You can use them to target and neutralize specific pathogens. They're transferable between humans. There are few to no stories about taking an antibody from one human and having problems in another human. It's almost impossible. And that again is because we have checks against autoreactive antibodies. There's a caveat there: when people start to develop antibodies in silico, you don't actually have that check, and so those need to be treated a little more carefully, in my opinion. They can be rapidly scaled and mass produced. We have systems in place for mass antibody production. The highest grossing drug in the world is a human monoclonal antibody. It's Humira. It's, I think, a $20 billion annual drug. And then beyond that, if you just kind of start going down the highest grossing antibody drugs in the world, I think it's like the top 20 drugs in the world are mostly human antibodies. So we do have large systemic ability to produce these rapidly, cleanly, and have them be stabilized for injection and treatment. Let's see. And then a history of consistent, reliable therapeutic use. The other way that we know these are quite usable is that there are people without humoral immune responses. They can't make their own antibodies. Every 3 months, they get an infusion of plasma taken from the general population that has, much like the mother's milk, exposure to local antigens. And that infusion of plasma alone prevents them from getting local infections. And so that's used clinically and widespread clinical use there. Okay. Any questions, guys? All right. So this is going to kind of go a bit outside of the immune system, but through the lens of an immune system. And so I'm thinking about how SARS-CoV-2 has breached our barrier system. And so much like the immune response, we have a barrier system. And how we can strengthen that barrier system is really the theme of this talk, and ways that we can do it scientifically and get ahead of a virus or a pandemic. All right. So current methods of plague management and where we've won: water treatment, widespread chlorinated water, soap distribution. I forgot to put that up here, but there's a great study showing that the mechanized production and distribution of soap may have been the primary reason why we had a reduction in communicable diseases worldwide. Not antibiotics, soap. Inspections and testing of food supply, that's a major barrier we have. Isolation, rapid isolation of infectious groups. Historically, you can go back and look at things like leper colonies. Ability to test for the disease. We've been scrambling to do better here, but could you imagine if we couldn't test for a disease? If a disease was, you know, in the ether and no one actually understood how it spread or who had it. The sheer existence of a World Health Organization and a CDC are barrier systems, ways to coordinate information and rapid responses. Again, we haven't done well here, and we've won by having it in place. We haven't won by using it effectively. And then continental distancing and rapid local responses. And so there is something to be said for why we don't have Ebola in this country, and it's because there's a continental flight between us, and you can have local rapid responses on the ground, which were done quite well in the prior administration, and the reason why we never had Ebola really leap to person-to-person spread in the US. All right. This is where I believe we've lost.
Wait a minute.
So a lot sir, of course, yeah.
I mean, I wouldn't go too overboard on the politics of Ebola, because Ebola is a very different disease that was transmitted in a very different way. If Ebola had been transmitted by aerosol droplets, it would have been a whole different story. It was transmitted by touching the blood of someone who was dying from Ebola, and that's not a common occurrence.
Yes, yes, I agree with that. Blood droplets too, though. So any droplet aerosolization, be it mucous membrane or blood, but I agree. I don't want to go overboard into the politics there. It has a more difficult path of spread. So that is true, but the spreads were, what I was excited to see was that our prior, and let's not make it political, I agree, but prior responses to Ebola were on the ground in Africa, MDs, medics, and trained epidemiologists on foot in Africa, controlling the disease at the site. And I think that's notable as a version of a barrier function, which did not happen in China for various reasons, I'm sure. Okay, this is where we've lost. This is how I think that we had this virus breach our barrier systems. And please do, I love open discourse on this. This shouldn't be a monologue. So any opinions here, I'm happy to hear out and discuss. So we have an extremely high population density, more so than even bubonic plague times, much more. We have intertwined trade networks. Speed of travel has increased. I think that people have done a poor job of educating most of the population about disease spread. The idea that people, have to be reminded to wash their hands is it's rough. That's, something that you learn first and foremost when you're working in a laboratory. Don't touch your face, wash your hands, everything is infectious. And people just aren't trained to think that way. And I think it's because we have a bit of a complacency in a time with no memorable plague, which leads to people who don't believe in science or don't really believe in vaccines. And I think, as a population, we could do better. Other ways that we lost to SARS-CoV, it's a high virulence virus. Mutations have indicated that it may have about 1,000-fold higher binding strength to cell surfaces than our prior SARS, was it SARS 2003? It engenders extreme differences in antibody production. This is based on a paper that was posted on bioRxiv 325, and I'm happy to send it out to the group if needed. But they looked at 10 different patients, and they found one patient that produced a good neutralizing antibody. And even within that one patient, there were a variety of antibodies that were kind of neutralizing, but not perfect. And so something about this virus isn't engendering a good antibody response amongst the entire population. To me, that looks genetic, but the jury remains out there. There's a long period of virus shedding before symptoms arise. Influenza, people often feel quite sick very early in influenza and don't persist in spreading the disease. If influenza had a 10-day period of time where people felt perfectly healthy, it would probably have a higher spreading profile because people just wouldn't naturally self-isolate. Asymptomatic carriers appear to be the dominant disease profile. The majority of people who have this virus, or there are many, many people, and maybe I shouldn't say majority because we're still having numbers change, but there seems to be a significant portion of the population who's infected and can continue to spread this virus without knowledge of being infected or being infected with anything significant. So that's very, very dangerous because the virus can persist in spreading unbeknownst to the local population until someone pops up and is quite sick. And then prior exposure to endemic coronaviruses does not appear to produce any sort of partial immunity. This is a great strategy the immune system has. Even if an antibody doesn't have a really high binding strength to a particular protein, it only partially binds, that will still buy time for the rest of the immune system to catch up. And so we see this a little bit with influenza. Sometimes people get the flu really bad, sometimes they just kind of get it. And that's often attributed to the idea that you've been prior exposed to influenza, some of the antigens remain the same, and you have a mild enough or not a mild enough, but you have enough of an antibody binding affinity to give the immune response a little bit of time to catch up. And so the coronaviruses don't appear to have any cross-reactive antibodies in the population. So that's not, going to buy anyone time. All right. Thoughts or questions, guys? Okay.
Question.
Yeah.
Sorry. Is a possible reason for the prior exposure to coronaviruses not conferring immunity to SARS-CoV-2? Is that because of this so-called spike protein, which is it's not only the differentiating factor of the new coronavirus, but it's also the main thing that any antibody could attach onto, and it happens to be different than what's been seen before, even though the internal proteins of the virus and the RNAs are quite similar. They're not available to be seen by antibodies until the virus infects the cells and the cells start presenting the proteins.
Exactly. That's exactly right. And so the thought is that even though there are some similar features, well, there's a two-part answer to this. So the thought is because the coronavirus is quite novel, and there are some similar features, however, not enough to have overlapping antibodies confer partial immunity. And that's been, I saw a research article go by where they looked at patients or people who had been infected with coronavirus. They're quite endemic. They usually don't cause anything more than a mild cold. But they couldn't find any cross-reactive antibodies in this paper. It doesn't mean they don't exist in its entirety. They just didn't find any in the first paper I saw on that. Second, we don't actually make the best long-lived antibody responses. We can make antibody responses, but it seems that to the SARS virus, at least, another kind of quite virulent coronavirus, patients, about 50% of patients at 3 years no longer had circulating antibodies or an antibody immunity to another coronavirus or to SARS, for example. And so the long-lived response that you would see in influenza or tuberculosis or something like that, if you have a vaccine, doesn't seem to be happening with natural infection of coronavirus. Okay. All right. All right, so this is my little reasoning speech on why I think coronavirus is here to stay. COVID-2 is here. As I said, many patients do not produce effective neutralizing antibodies. That means that it will continue to circulate. Coronavirus immunity does seem to fade after 2 to 3 years. And this is based on SARS from 2003, studies there of those patients. High infectivity offers more chances for the virus to mutate. And so if we have a 1,000-fold increase in the spike protein binding, we have wide distribution across the population, many people who are asymptomatic, this gives the virus more chances to mutate and develop different surface proteins and escape any natural herd immunity that we do develop, even though there doesn't seem to be a lot of pressure for the virus to escape that at the moment. And again, the successful maturation of the virus to escape natural herd immunity, if that happens because we have widespread infection, it will become a bit more like influenza. This is my personal opinion. I'm not referencing any articles here other than my knowledge of the immune system, and I just want to caveat that. Okay, often viral mutations. Typically, we kind of get a break as a species. Viral mutations often trend towards lower virulence, and that's because it increases the viral spread, and that virus will outcompete the original high virulence virus. There just isn't that developmental pressure here with SARS-CoV. It's quite competitive, it spreads quite easily, and people aren't producing good immune responses to it, so there's no pressure for it to mutate and towards something of lower virulence with a higher spreading or whatnot. And then, of course, the longer shedding period of asymptomatic cases. All right. So, any questions on that? That was a bit of my opinion. This is how I'm thinking about things. I'm happy to discuss with anyone.
Melanie, just one question from before. Is the asymptomatic spreading, is that unique to COVID, or is it just more pronounced?
I'm sorry, I missed the first part of your question. There was a bit of a drop.
Is the asymptomatic spreading, is that unique to COVID, or is it just more pronounced?
So I think that's a really good question, because I'm trying to remember if I've seen any SARS research on that. Because coronaviruses are typically quite mild, we don't test people for them. We say, oh, you have the common cold, go home, you'll be fine in 3 or 4 days. So I don't know if there's good information about that. That's a good question. And I think the best place to look for that would be any of the SARS 2003 epidemic research to see if there was asymptomatic spreading.
I have one question as well.
Of
What
course.
does it mean if immunity fades after 2 to 3 years? If you get it again, is it as bad as the first time around?
That is the idea. That is the thought, that after 2 to 3 years, you do not have plasma B cells left that would recognize the virus, and you would start over.
That's crazy.
Yeah, it's not good. Yeah.
I have a question. What do we know about the 3 strains and whether there's any possible cross immunity?
Yeah, so I have not, this is on my to-do list. I have not started to look at different strains. I've heard as many as 20 being isolated. There is likely going to be cross-protective immunity, as massive mutations typically tend to make a virus less virulent or inactive. And so you would imagine that there might be some cross-protective immunity between these mutations. However, it's not guaranteed, because the way the immune system chooses an antibody is, okay, we've got the best one, let's go for it. If that best one antibody is to a part that did mutate, you can get escapism of the virus from herd immunity. But yeah, that's definitely on my to-do list, is to start analyzing the mutations that we've seen. And I think that's actually a critical part of the response.
So on the last point, where you're saying that you don't think there would be a strong selective pressure towards lower virulence because it already spread so easily.
And because we have asymptomatic cases, so it looks like a lower virulence.
But you can imagine that it could just be even longer. It could just have an even longer asymptomatic period, in which case there would be a selective pressure towards lower virulence in spite of this.
I'm trying to follow your argument. Can you repeat it?
So just because this virus has a long period of asymptomatic spread, doesn't mean that it could be even longer, right? You can imagine this just staying with you for a month, where so maybe there could be a selective pressure for it to be even less virulent, even less detectable. In which case there is a hope that that the future versions of this virus will be less deadly.
Yeah, that's an interesting thought. I don't believe, because you have asymptomatic cases, I've heard of people still testing positive for up to a month with this already. So therefore, these are people who are walking around, they feel fine, they're in isolation on exercise bikes every day, they're not experiencing symptomatic responses that would indicate that they have the virus or they're carrying it. And that's part of why I don't think there's a strong selective pressure for the virus to escape via mutation. It doesn't need to mutate to keep infecting people and have a productive infection and spread elsewhere. But these things are not immune from randomness, and viruses mutate all the time. It's just which version is going to be dominant. It's going to be the one with the least immunological pressure to mutate and greatest ability to spread, and I think that's what we're seeing here.
Do we have any idea what the prevalence is in humans or any species of generally asymptomatic viruses? Like, why would we even care if this virus was largely asymptomatic in everyone?
No one would notice.
So do we have any idea if that's a thing, that there's tons of viruses going around that are largely asymptomatic and just have sort of reached a homeostasis with the host species?
Oh, absolutely. And in fact, there's a book written by, it's a little bit dry, it was written by a prior advisor of mine called Darwin's Blind Spot. And the premise of the book is that viral infection is actually co-opted, or rather co-developed along with our evolution. And so they point to snippets of viruses that are throughout our immune system and in our cells where they've actually integrated and conferred interesting features and functions. And so the idea is that we've evolved more rapidly because of coexistence with viruses that don't kill us effectively. There are many, many viruses that are endemic to our population that we don't really notice. No one would care about HPV if it didn't induce cancer. Many people are walking around with that. I think it's like 90% of the population is asymptomatic infected. And there are other viruses like that.
I have a question about the mechanism of the invasion of the virus.
So in
a way that has, so it has to bind to a receptor on the surface of the human cell, and that receptor doesn't mutate that fast. So there has to be some kind of conserved domain in a virus, spike protein, that will always allow to bind to the human cell. Why can't a human immune system produce an antibody to bind to that part, which is not supposed to mutate that fast?
Yeah, that's the goal. So that would be the ideal antibody, one against a portion of virus that cannot mutate and still have the virus be successful. And that would be a great broadly neutralizing antibody. Antibody generation is difficult, but that would be the ideal one to search for. That or a stalk protein that neutralizes the virus and is something that would not mutate. The human immune system can build an antibody to that. If and when it does naturally, then that's immediate pressure for a virus to mutate. And so you could imagine if you have a billion viruses in your system and they have a mutation rate of a given set, the ones that go on to infect other cells are going to be the ones that no longer have that antibody binding. So mutations can arise. Antibodies are so specific that if you have one similar amino acid change in an antigen profile or in the antigen profile that doesn't alter the way the protein functions, it will no longer have a good binding affinity with the prior antibody. They're extremely specific, and so that is what you would want ideally. It does allow for selection pressure, but it may be the best protective therapeutic we have.
Thank you.
Mm-hmm.
I have a question for you on the vaccines and some of the implications. My soda straw is looking through humanitarian action, so displaced populations that are in great risk from this particular virus. One of the first interventions that we usually do is to give a measles vaccine. This is something where its spread could decimate a refugee population very quickly. Is the vaccine, given 2 to 3 years of immunity from even catching this, going to provide that level of protection? Or are we looking at a fundamentally different type of pathogen and going to need to adapt our systems?
This is a really good question. And so the coronavirus immunity fading, this is natural infection. As far as I know, there was never a SARS vaccine developed. Vaccines often lead to, or can lead to, longer memory-based protection because they come with a lot of inflammatory factors, adjuvants, and the immune system really has time, because you're not doing it, typically not doing it with a live virus. The immune system really has time to get its arms around that and develop the best possible antibodies. I think it's still possible that we could develop a vaccine that would confer protection for longer than 2 to 3 years. Those numbers are based on natural infection, not adjuvant boosted infection like you see or adjuvant boosted antigen exposure like you see in vaccines. However, though, it is worrisome that you don't have anyone really, or large populations developing coronavirus immunity. There may be something specific to the coronavirus that makes that difficult for our immune systems to do. That's my best answer on that topic.
That's frightening from my perspective, but yes.
It is. Yeah, this is a big question, and I think we're going to do some very rapid. I think we'll do some very rapid vaccine development. Fingers crossed we get something that works 9, 12, 18 months from now. But after that, I think, you know, if coronavirus is staying around and we've figured out ways to mitigate and manage it, there will be a concerted effort to look for a long-lasting vaccination, or, you know, we get it every year like the flu shot. But it's true that distribution and flu shot vaccines are difficult to get to everyone. All right. Any more questions, guys?
Oh yeah, I thought you were going to talk about were you going to talk about manufacturing antibodies outside of the body, or did I get a misimpression?
Yeah, we're going to get to that, and I can skip ahead if we're on. How are we doing on time, Allison?
Well, it depends on how much time other people have, but how long is the presentation still from now on?
Let's give it a shot. Let's see. So I was going to go over. I can go over some things pretty quickly. Yeah, I'll get into it in about 2 slides. If I just 2 or 3 slides. Does that sound all right, guys?
Yep.
All right. Yeah. I can also go a bit over time. You know, so.
Yeah, I'd rather see everything, even if it goes overtime, than to have to skip anything.
I agree.
Okay, great. Thanks, guys. Allison, maybe we extend the time on the Zoom so it doesn't cut everyone off.
Yes, I have. This room is always open.
Okay. Okay, great. Very smart. Okay, excellent. Okay, so this is how I think about our standard immune response, how to hack it. So we have corollary to the immune response. We have things like water treatment, food shortages, or rather food inspections. Can you guys see my cursor on this? These slides are giving me a little complicated.
Yep, we can.
Okay, great. Thank you. And then for the innate, we have a cellular broad response. This looks a lot like broad therapeutics, hospitals, things like anti-inflammatories. And then there's things like targeted therapeutics, which look a lot like the adaptive immune response. And what I'd like to propose, a way to get around this plague and around other plagues, especially given what we know about its virulence, its continued spread, and its potential persistence in our population, is that we start thinking about something as a 4th branch of the immune system, which is physical external. And this means preemptive adaptive immunity. And how do we get there? And really taking a hard look at societal responses and responsibility in things like barrier protection and things like broad therapeutics and hospitalizations and being prepared better for something like this. But really, here we go. I'm gonna close something here. But really, if we can use the science we know and how we do it and producing antibodies a priori in a dish, we can do something really novel here and something that I think would be a mass benefit, which is have our adaptive immune response move to the forefront as a physical external mechanism for preventing pandemic plague. We have enough computer science to predict what those mutations could be. We could have antibodies on deck to inoculate populations as soon as we identify a novel virus. And, you know, really, this is a way that you can prevent mass pandemics, shutdowns of governments, etc. And this is how I think about externalization of the adaptive human immune system. If we don't have to wait and to see if people are developing a proper antibody, X number of people are infected. Okay, now we have to go get that antibody from their plasma. We have to analyze it. We have to transfer it to other people. If we had antibodies on deck, which can be stored for a long time, they can be stored in sequence format and spun up in 4 weeks for cGMP production. We have a way to get our arms around preventing any plague in the rest of time. And so this is really what it looks like after you get infected. This is influenza, what happens to you. So you have your initial innate immune response, which we've gone over, cytokine storm, which is responsible for local inflammation. You have your adaptive immune system, your T cells start to move and do things about, I've seen earlier than this, maybe 5 or 6 days early, is the peak of T cell response to influenza. And I have a research article on that if you'd like to read it. And then you have your antibody production really happening throughout this at some low level, but this very specific antibodies take about 10, 12 days to kick in their production. The idea here is that we move specific antibodies to the front line and use them as part of our external adaptive or innate immune response as a barrier system. All right, and this dynamics of herd immunity, I just wanted to touch on this in case people weren't familiar. You have several infections over the period of your life, and you develop a repertoire of antibodies and abilities to respond to local infections. We would be moving this repertoire to the forefront, and this would be through antibody production and injection, perhaps specific ones to plague, perhaps antibodies to a specific virus like COVID. We could potentially eliminate it from our population by introducing temporary herd immunity for an extended period of time of a couple of years. We could drive this virus back to the animal kingdom because it's no longer transmitted or infected between humans. That's what we saw with polio. We have very, very few cases of polio in the world, and that's what we saw with smallpox. We're able to develop such herd immunity that these viruses are no longer considered endemic, and in modern medicine aren't worried about as much. Okay, so that's that. All right, and so this is my take on antibodies. I think they're a flexible, fast, modern panacea. We can produce these. Our company, Prellis, full disclosure, I'm the founder of this company, can recreate human immune responses outside of the body. And the way we do this is by recreating the physical structure of a human lymph node. That is actually really critical. As I said, looking at how cells move through tissues was my research for about 15 years. Having that three-dimensional physical structure that guides cells to respond and do the guess and check work is actually important for developing the best possible antibodies. And so we were able to recreate that in our time as a company at IndieBio. It was the first project we did because it's really what I know best. And in 2 months, we had anti-human Zika antibodies. We had antibodies from 3 different donors. They'd never been exposed to Zika. They were totally healthy blood donors. We were also able to recreate this against H1N1 and a protein called CTLA4, which is a cancer checkpoint inhibitor. We were able to get human antibodies against a human protein. So we are able to do this in a dish, and we're able to do it in a matter of weeks. And I misspoke. I said 8 weeks. That was to the full testing and characterization. But 4 weeks in, we had positive human antibody hits. And so, as a small company, we thought this was great. It was a good demonstration of our technology working quite well. And people really liked the story that we could produce organs and tissues that would be transplantable. And so that's what we pursued. Although we did file plenty of IP on this, and the patent was actually issued in December of last year. So quite timely towards the end of December there. All right. So human antibody production, there are a lot of ways to go after it. The current methods are a little bit flawed and take a little bit too long to do what I'm talking about, which is move the antibody into the barrier system of human disease. Right now, there are a couple companies, and I'll review them. They look at human infected or patients that have been infected. So there's human infection required. This isn't ideal. You have to go after groups of people who have encountered a novel virus. You have to get their blood, you have to screen it. They may or may not have made good antibodies to it. That's a long screening process. You have to do a lot of sequencing. And you really, you have to have feet on the ground working with dire infected people. It can work. It takes a while. I don't believe it's fast enough to get after a novel virus or a plague. Again, intensive screening processes are required. Most require 3 to 24 months to produce antibody hits, and they're quite expensive. This isn't something that you're going to want to do over and over again, and it's not something that lends itself well to developing an antibody against an a priori infection. This is a very dense slide. Happy to distribute this later, but generally there are some people doing this. Regeneron's using human B cells from infected patients. Takeda is using pooled human serum, which may or may not contain enough neutralizing antibodies. They're running a clinical trial with that quite quickly. As we know from the Bioarchives paper that came out of China on 325, again, happy to share that later. Not every patient is making neutralizing antibodies, and not every patient is even making a lot of effective neutralizing antibodies when they are making them. Okay, and then distributed bio, one of the founders, Jake Landville, is a great guy. We happen to share an investor, and they do a lot of in silico antibody modeling and silico modeling to get after a universal vaccine. And I hope technology works. I think they've got a good shot at it. But one of the critical components they're missing with in silico antibody generation is that autoreactivity component that Kriyan asked me a question about earlier. Our bodies automatically eliminate autoreactive antibodies as they do autoreactive T cells. That happens during that guess and check process. And there's whole mechanisms defined on how that's done. But if you develop an antibody in silico, you may have cross-reactivity to human proteins that's unanticipated and really difficult to screen for given the entire proteome of the human body. And so there could be unintended consequences of going really fast with a technology like that without really checking. Okay. So this is a picture of one of our little mini lymph nodes. That's over there on the left. And that is a set of different immune cells that we grew up in the lab from a human patient blood draw. One blood draw, we make about 500 of these. We lit them up in different colors for this picture specifically, usually they're clear, because we wanted to highlight different zones. And so the red zone there is where our B cells and likely our plasmacytoid DCs are hanging out. The green zone there is where we have our T cells, our CD8s, our CD4s, our regulatory T cells, our follicular dendritic cells, and our regular CDCs, our regular dendritic cells. And this is something we did again in 2017 in a 24-well plate. We had 11 active Zika antibodies produced. We run a numbers game with this. Statistically, as I mentioned, the strongest B cell wins. And so the B cell getting the most signals and positive signals has the strongest antigen binding force to that dendritic cell or that T cell. And that kind of tells that B cell, okay, I'm the one that needs to go and divide. This is very Darwinian. The ones that get the strongest signal go out and divide and proliferate. The ones that get weaker signals don't proliferate as much. And so if you run a numbers game, a statistical numbers game against how many naive B cells will you have in a dish, how many times do you need their receptors to divide, and how do we think we're going to win, you can start with a certain number of B cells to get a dominant B cell clone in each dish. And that's what we did here. And that was one of the pieces of the puzzle that we solved. And so that way we make sure that we get one strong dominant B cell in each dish. We go ahead and we screen the antibodies. We pick the positive hits. B cells have this really great feature where you can immortalize them. And I can talk a little bit more about hybridoma creation. It's been around since the 1970s. It's been demonstrated with human B cells. We did it in the lab. Hybridoma is you fuse a B cell that's producing crazy amounts of antibody, that plasma cell, with a cancer cell. And that cancer cell immortalizes that antibody production. And so you can do that, and then you can also sequence the antibodies and then immediately start scaling them for cGMP production. But before cGMP production with the antibodies, hybridoma methodology was the way that most antibodies are made and the way that many are made today. So really being able to get after a group of dedicated plasma B cells producing the correct antibody is a feature of our technology that allows for great speed. Do you guys have any questions? That was quite a bit.
So are you doing this now for Corona?
Yeah, we're doing this now. We registered with the CDC last week. We'll be getting antigen in-house in about a week and a half, and we're getting our first blood draws from UCSF on Tuesday.
Great. So any idea how long you think it'll take to work? You think it'll be like 4 weeks or?
Yeah, so our estimates based on what we did prior, it should take us about 4 weeks before we have our sequences in hand. Yeah, and that's 4 weeks from the day we get the antigen. We can't start without the actual viral infectious component. And so it should take us about from today, maybe 5 or 6 weeks tops. And you know, there's always biology involved, so we may have to go at it a couple times, but I think we're going to parallelize several donors and try to have this done in a few weeks.
I have a question. Does your strategy rely on just using one or a couple plates and selecting the best, or do you mimic this sort of mutate and recombine and try again strategy to create more and more effective antibody producing B cells?
Yes, and so yeah, that's a strategy we leverage. In each plate, in each well, that's the immune response we set up. And so and this is one picture of what's going on, but about 4 or 5 days post vaccination, as we call it, you know, we give them the antigen, we give them a booster, it looks a lot like a vaccine milieu. We actually see the B cells doing their coagulation, their division, and their movement back and forth to the different zones to interact with the T cells. And so we actually set that process up in a dish, and that's what we're leveraging.
Will this work as like an anti-cancer vaccine as well? So.
Yes, one of the antibodies we went after was a checkpoint inhibitor antibody called CTLA-4, and we developed antibodies to that as well. So this technology is flexible beyond viral pandemics. It's really a great way to just generate a human antibody rapidly and to leverage the human immune system outside of the body to do so.
That's really good. Amazing.
Thank you.
So I have a question. So this is David. I have a question. There was some effort that got some publicity about 10 or more years ago, led by Nathan Wolf, about sentinel screening near, for example, bushmeat markets, where zoonotic viruses that had never been characterized could be identified. And the idea was, what's it worth to the world to identify HIV, characterize it, and start researching the biology of it before it really has swept through a few continents. Yeah. And I'm wondering whether you have, in your more relaxed moments, a dream of a larger, ongoing, sustainable scheme for defense against these little vicious nanoparticles.
Yeah, absolutely. And you know, the question of what it's worth to the world, it's probably hundreds of billions of dollars. Yeah. So what it's worth to the world is significant, and yes, that is something we are looking into participating in. Before this pandemic even started making headlines, we'd actually submitted a proposal to DARPA on these lines and have met with them and are discussing longer term projects that are involved with things like you mentioned, sentinel monitoring and looking for novel viruses where they may emerge.
Thank you.
Okay, all right.
So
Anyone
then
else?
you would need, yes, one more question here. You would need to produce this and inject it into everybody regularly because they cannot produce it themselves, as in the vaccine case. So you would need very large quantities and good distribution. How much would it cost?
That's quite the question. Antibody production is relatively well understood and fast, and can be done quite inexpensively. You know, I foresee this as being, you know, while this virus is endemic, before we can kind of push it back into the animal population would be our goal. I would foresee, you know, 4 injections a year for as many people who might be exposed or vulnerable. And I can't really intuit the numbers on that. It's not inexpensive, but combined with good screening and testing and isolation plus antibodies, I think it could be, you know, it may not be something we need to do for the whole population.
In an ideal case, if everything happens according to plan, could you give us a timeline of what will the next month, what would the next years look like if what you're working would get the funding that it needs and could be rolled out in the way that is an ideal case? Yeah,
we're also talking to pharmaceutical companies for partnerships because we think this could go a lot faster, but I think we could have something that's scaled up for GMP production by August or July. And I think from there, depending on the motivations of the people involved, we could have it for distribution in the fall. That would be ideal.
And how would distribution work, and how fast could it be rolled out across the globe, and what would happen? What would
need to happen? You know, we'd probably look at different ways to distribute it as fast as we can. Once we've got an antibody that we know works, distribution can happen as fast as sending a sequence and getting the people on the ground wherever they are to scale up production. Antibody production happens all over the world. It happens in Europe, it happens in Argentina, it happens in the US, it happens in China. These facilities and places are ready to go. And these, you know, it could happen quite quickly and be localized and distributed locally. And I think that would be the fastest
And
way
how
to do
could
it.
people, okay, how could people here help if at all?
Yeah, I'm delighted to talk to anyone offline about what our investors are looking like. We've been doing a lot of investor meetings, as you might have imagined, the last couple of weeks and this week. I think our current investors are probably going to just step up and fill what's going on. But if there is any room, you know, I'm happy to discuss this with new investors who can help us make this go faster.
All right, thanks. Any other questions?
Can you speak to the possibility of ADE? I know that was discussed in some of the early research on SARS-CoV-2.
ADE, can you define that for me? It's not coming to mind, sorry.
Antibody dependent enhancement, like what happens with dengue fever, where the second infection.
Yeah. So dengue fever is an interesting one where natural infection, the first time you're pretty darn sick. The second time around, the antibodies that people tend to make actually make the infection worse the second time. It's one of the, I don't know of another good example. Dengue is probably the most popular example of this happening. As far as I know, that has not been demonstrated for a coronavirus or an influenza virus. Doesn't mean I don't think it's impossible. I think that if you have a broadly neutralizing antibody, which we can test in vitro, or an antibody that does neutralize viral entry into the cells, it would be atypical for it to also cause better infection at the same time. And that can be screened prior to patient introduction. And it would be part of it is part of our stated screening process where we look for true viral neutralization. Typically doesn't make the virus more virulent, but that does happen with natural infection in dengue. Thank you for pointing that out.
I have a question. Will these antibodies be injected or introduced intranasal? And do you plan to use any adjuvants or preservatives for them?
Yes, so they'll likely be injected. If they're being used therapeutically, someone's already infected with the virus and they need a way to calm down the immune response and buy some time. That would typically be an IV infusion. You can have it injected subcutaneously. Intranasally is interesting. You know, there are a lot of studies about trying to introduce protein therapeutics into the lungs. I actually spent some time researching one of them from a company, ancient history, they're long gone, called Inhale, where they were trying to introduce insulin through the lungs. This introduces idiopathic pulmonary fibrosis in a lot of people, and it's really not a good way to go after antibody absorption, especially if you already have a lot of mucus and inflammation in your lungs. It's probably just better to inject it. And preservatives, there are standard antibody preservatives. I don't believe you would ever want to put an adjuvant with this. You don't need to activate the immune system for these to work, but you would have standard protein preservatives alongside of this, such as human albumin, which we already have 60% of our blood volume, roughly 50 to 60% of our blood volume is human albumin.
So in your first slide, or maybe your second or third, you said kind of that the world is now significantly changed. And even in the best case scenario, if we could do all the things that you're proposing, and if this actually worked, and you got the funding and everything was rolled out, what do you kind of like foresee other things to be that people would still need to do? Like, is this kind of like the kind of like the golden shot against everything, or what are other kind of like changes that we would still need to implement?
Yeah, that's a really good question. And so I think we need to pay strong attention to, and I'm sorry, since I've marked my screen, I'm not sure how to get rid of it. Let me see if I can, no, okay. I think we still need to pay a lot of attention, Allison, to the physical barriers, things like water treatment, general monitoring of sentinels, standard ways that we as a population can protect ourselves from rapid pandemic viral spread. We've gotten lax about it. We haven't seen that. This is the first plague in living memory. And I think that, you know, a lot of people will be thinking very, very hard about how to do this. And I think that in addition to ways to respond to viral plagues when they do happen, because they will happen, and ways to try to get our arms around this one, are really to use the power of science, perhaps antibody development, as we're proposing, move this into the early innate response where we stop viral pandemic spread in its tracks.
All right, thank you so much. Is there one last question of anyone? Okay, thank you so much for staying on. And oh, yes, go for it.
Hi, Melanie, this is Milad. I have a question about the mass production of this antibody. Let's say that we found the antibody that works with these pathogens and everything. What's the right method for the mass production? Can you talk about that?
Yeah, so it's going to be good manufacturing practices, FDA certified. There are probably about 50 or 60 companies in the US that do this already. One that we're looking at talking to is SwiftScale. If anyone has a contact there, happy. I know they're very, very busy right now, but we'd love to chat with them. And what they do is they take either the sequence or the hybridoma and they express that sequence in a mammalian cell. And much like the way we secrete the antibody in the serum, the cells also secrete the antibody. And then they gather the serum, it's processed on site, and they produce a GMP product. This is actually really done almost every day throughout different supply chains. And it's extremely well worked out, almost boring manufacturing process at this point. So we would provide the sequences and perhaps even the hybridomas as requested, and we would allow a different facility to do the cGMP manufacturing.
Thank you.
You're welcome.
All right. Well, thank you, Melanie. I mean, I think it is really a testimony to your efforts right now that people were willing to stay on 20 minutes after, and we even, I think, got some more people to join. So probably people rallied some troops. Thank you so much for taking the time to give us this really fascinating, terrifying, but also hopeful outlook.
Yeah.
I think that, yeah, if you're willing, I will share the slides on the schedule. Maybe you wanna put in your email address or another way that people can contact you right now in here, just in case.
Sure. So my email
okay.
address is very simple. It's at the bottom of the screen. It's my first name at prellisbio.com, and I'm happy to answer questions. And like I said, I'll share any references that I was talking about in the talk.
Yeah, one last question.
Right.
What was the name of that company? The audio cut out when you said it. The manufacturer.
Oh, SwiftScale.
WitScale?
SwiftScale.
Got it.
So they apparently have a way to scale cGMP antibodies in a matter of weeks, and we'd love to talk to them.
All right. So I shared a little bit more info on how to join those salons and what they are and so on and so forth. In the interest of time, I'm not going to go through all of this, but in case you want to join a future salon, please go for it. And Melanie, in case you have any updates on your research and how people can help, please reach out to me, and I'd love to do a future salon. That is, I think, one of the most succinct summaries with kind of like with a hopeful outlook that I've seen so far of the situation. Thank you so much for taking the time this morning, and yeah, I can't wait to be in contact again. Thank you.
All right, thank you, Allison. Thank you for having me, and thank you everyone for staying extra.
Thanks, Melanie.
Yeah, thank you guys. I'll share all the info on the Sanity schedule. Bye everyone, have a lovely Saturday, and I see many of you tonight here at 6 p.m. with Mark, Christine, and I doing something on voluntary cooperation versus top-down control to fight this crisis. Bye-bye.
Bye-bye.